Good afternoon, everyone. Welcome to the sixth problem solving session for the course Waste to Energy Conversion. So, I am one of the TA for this course. My name is Asma Parveen, and I am a sorry, and I am a PhD scholar in Civil Engineering Department at IIT Kanpur. So, in week 6, I'm so sorry. So, in week 6, there was 5 lectures. I think the first three lectures was on anaerobic digestion. The fourth lecture was on microbial fuel cells, and the last lecture was on fermentation. The first part of fermentation was in week 6, and the second part will be in week 7. So, what we are going to do is that first we will see a brief recap of week 6 lectures, and then we will move to the question. So, let me share my screen. So the first lecture of the week 6 was on energy production from organic waste through anaerobic digestion. So we have seen different pathways for energy production like I'm so sorry, combustion, pyrolysis, gasification. Now we are looking for the anaerobic digestion. So first, what is anaerobic digestion? It is a biological process in which microorganisms break the biodegradable organic matter in absence of oxygen into uh, gaseous product, and the left and the sludge or the biomass is converted into some sort of compost. So the feedstock which can be used for anaerobic digestion is like agriculture waste, some of the energy crops. So these are the I think some type of grass which have very high calorific value. So when they are digested, they give very high yield of biogas. Next is the animal waste. So animal waste like cow dung, it can be digested to produce biogas, and also there are already lot setups for biogas production from animal waste. Next is industrial waste. Next is municipal solid waste, like food waste and other waste can be digested anaerobically to produce biogas. Again, restaurant waste and also sludge from wastewater treatment plants. So there are various STPs in India which are digesting the sludge anaerobically, and the biogas which are produced from the digestion of the sludge is used to run the plant for some time. So the primary end products which we get from anaerobic digestion, first is the biogas which has high calorific value. Next is the hydrogen gas which will be in the gaseous form. Next is the biofertilizers. So what happens during anaerobic digestion? If whatever organic product is there in the feed, it is digested and converted to biogas, but this liquid supernatant is very rich in nutrients like nitrogen and phosphorus. So when this supernatant from the digester is applied on the fields, then it gives very high yield, and it can replace a small some proportion of the artificial fertilizers which are added in the fields. Next is the solid part which we get from the anaerobic digestion is the compost. During anaerobic digestion, the sludge or the feed is converted into the compost like material, which has very high nutrient content, like it has a high nitrogen, phosphorus, magnesium. Also it is free from pathogens, and as compared to the initial feedstock, and it can be used as compost in the fields. Next is reusable water. So the supernatant from the anaerobic digester, after its proper treatment, it can be reused again. So let's see what is biogas. So biogas is a combustible mixture of gases. It has high calorific value. It mainly consists of methane and carbon dioxide, in which CO2 has lesser calorific value than methane. And it is formed during the anaerobic bacterial decomposition of organic compounds in absence of oxygen. So if we see the major components of biogas, it contains of 50 to 75% of methane. CO2 is 25 to 50%, and we can see that along with methane and carbon dioxide, there are some other gases which are formed, like hydrogen sulfide, and oxygen. And these extra gases which are formed, these are act as contaminants, and they need to be removed before using of the biogas, because they causes some of the operational problems, like it can cause corrosion, or it can if there is dust in the biogas, then it can choke the equipments used for energy generation. So next here is a table which shows the waste and their methane content, and along with the biogas potential. So we can see that all these waste have very high biogas potential and methane content, all is more than 50. Again, this is the continuation of that previous table. So here is a flow chart showing steps of anaerobic digestion. So here we can see that for pre-treatment for lignocellulosic biomass. So it has lignocellulosic biomass a very high content of lignin and is very hard to digest. So this some pre-treatment is required for that. After that, the complex organic compounds like carbohydrate, fat, and proteins, these are converted to simpler organic compounds like sugar, amino acid, and this step is known as hydrolysis. The first step is hydrolysis. Next is the simpler organic compounds. These are converted to short chain fatty acid. This is known as acetogenesis. Also, the simpler organic compound can directly be converted to acetic acid. After the formation of short chain fatty acid, it can undergo it can undergo two pathways. It can produce either acetate, or hydrogen producing acetogenic bacteria will produce H2 and CO2. So after acetogenesis, we have two products, acetate and hydrogen. Both acetate and hydrogen are converted to methane by two different pathways. So acetate can be methane converted to methane and CO2. Also, hydrogen can be converted to methane and CO2 by methane producing bacteria. So here is the pathway. So we can see that from hydrogen, only 18% hydrogen to methane route constitute around 28%, and acetic acid constitute around 72% of the methane produced in the biogas. So here is the elemental description of anaerobic digestion. So since it is a bacterial phenomena, it is dependent on microbes. So different factors will affect the biogas yield. Like there should be some optimum solid content in the feed. The organic loading should not be very high and should not be very low, so that the microbes cannot sustain. Next is there should be a optimum carbon to nitrogen ratio. The temperature should be optimal. It is generally around 35 degrees centigrade. The pH of the solution, so different, like acidogenesis, they require acidic pH, and methanogenesis, they require neutral pH. So pH of the solution should be optimum. And next is retention time. Since the anaerobic digestion is conducted by anaerobes, that is the anaerobic bacteria, so their retention time is required here is very high as compared to the aerobic bacteria, because their growth yield is very slow. Next is the type of the digester, and last is microbes type. So here is a group of the microbes which are used in anaerobic digestion. First is the non-methane forming, and other one is the methane forming. So if in the non-methane forming, there will be two types of bacteria. First will be the hydrolyzing and fermenting bacteria. Next is the acetogenic bacteria, in which by hydrolyzing bacteria, uh, some organic compounds are converted into acids, and in genetic bacteria, these converts the hydrogen producing, these are the producing acetogens and the homoacetogens. Next is the methane forming. So we have seen that methane can be formed from two products, that is hydrogen and acetate. There will be two class of bacteria. First is the acetoclastic acetate degrading bacteria, and next is the hydrogen utilizing bacteria. So the role of microbes involved in the three stages. So very first step is the fermentating bacteria, which converts the complex organic compound into simpler one. This process is known as hydrolysis. So we can see that all this cellulose, protein and fat are converted to short chain organic compounds like hydrogen and CO2. So we can see that the protein is converted to amino acid, which is further converted to this one. Fat is converted to fatty acids and alcohol. Cellulose is converted to monomers, oligomers and saccharides. So the next step is the hydrogen producing acetogenic bacteria, which decomposes the simpler organic compounds produced in the first step to acetic acid and hydrogen and CO2. And the last is the methane producing bacteria, which converts the hydrogen and acetate to methane. So here is some of the genes of the bacteria discussed, which produces the methane. So here we can see that the complex compounds are converted to these simpler compounds, like cellulose is converted to glucose and cellobiose. Proteins are converted to amino acids. So please go through these. So we can see some of the reaction by acetogenic bacteria, that is a hydrogen producing acetogens. Here the acid is converted to hydrogen, and we can see that propionate is converted to ethanol, that is acetate. Sorry, it is not ethanol, it is converted to acetate, and again we can see that here it is propanol, which is converted to acetate and hydrogen. So next is the methanogenic bacteria. So around 70% of the methane is produced by acetate, and these methanogens are highly sensitive and they have very slow growth rate. So if pH decreases or increases or something happens in the reactor, then the gas will be lower, and because the methane forming bacteria, which constitute around 70% of the methane, they are very sensitive, and they have very slow growth rate. Next pathway is from hydrogen and CO2. Around 30% of the methane is produced, and they are less sensitive and they have higher growth rate. So formic acid and methanol can also be converted to methane by bacteria, and the reaction is given. So these are the strains of the bacteria which can convert acetate and hydrogen to methane. So, I have discussed in the flowchart that some sort of pre-treatment is required for lignocellulosic biomass. So some of the physical pre-treatments are ball milling, that size reduction is required, irradiation and other methods. Then next is the physicochemical pre-treatments like explosion, alkali treatment, acid treatment, or with some sort of oxidizing agents are used. And last is the biological treatment, in which some sort of fungi are used or other microbes are used, which reduces the degree of polymerization of cellulose, or it leads to partial hydrolysis of hemicellulose. Or also the possible changes in biomass is, since lignin is very hard to digest, so delignification takes place. So let's move to the next. So the second lecture is also on the energy production from organic waste through anaerobic digestion. So here is the flow sheet for anaerobic digestion. So we can see that after pre-treatment of the feed, anaerobic digestion is conducted. We get the gaseous product, and all we get the digested effluent, which after dewatering we can get sludge and we can get liquid, which after proper treatment it can be discharged or it can be sent at the start of the treatment plant if sludge is being digested. So there are two types of anaerobic digester. It can be high or it can be wet. So if basically in dry anaerobic digestion, the total solid content is high as compared to wet. So in dry, it is 25 to 45%. So this much will be the solid and rest will be the water. But in wet, the solid content should not be more than 15%. So as in dry reactors, we are having high solid content, so it will be requiring less volume for the reactor. Solid to separation, liquid separation will be simple. Its energy balance will be more improved. It will be economically more viable. Its retention time will be shorter, and will have greater flexibility of feedstock to be feed. So here kinetics of methane production is discussed. So as we have seen in the previous lecture, so methane production of methane from acetate is the slowest process, and so methanogenesis is the rate limiting step. But if cellulose content is high in feedstock, hydrolysis may be rate limiting step because converting cellulose to simpler organic compound is very slow, and so if cellulose content is higher, then hydrolysis will be the rate limiting step. Also, presence of lignin decreases the rate of hydrolysis and further will slow down the hydrolysis reaction. Also, the transfer of gaseous product to the gas phase may also be the rate limiting step for some cases. So here our formula is given for theoretical substrate conversion rate per unit reactor volume, and this is the formula. Please go through this. Next, retention time is discussed. Please also go through these. So some of the facts for anaerobic digestion are given. So methane fermentation generally takes place at thermophilic temperature. So if it is taking at thermophilic temperature, then it increases the methane production rate, because generally it is seen that with increase in temperature, the reaction rates increases, so methane fermentation will be higher. Next is reduced pressure also provide little or no benefit. Low pH and short retention time precludes growth of methanogenic bacteria and thus reduces methane production. So methanogenic bacteria thrives in normal pH, neutral pH. So pH of the any feed should be neutral. And since it is a very slow growing microbe, so they require larger retention time for multiplying. Next is rapid continuous agitation of the anaerobic digesters is not necessary and in sometimes even harmful. So in anaerobic digestion, intermittent mixing is done, and sometimes some other maybe a mechanical agitators or some gas can be used for mixing. And if there is a vigorous mixing, then there are chances that these methane forming bacteria or the other bacteria in the reactor, they will not be able to multiply, and so it will be harmful for the digestion. And the specific methane yield, it is directly proportional to the biodegradable COD or volatile solid loading in the digester. If the feed has higher biodegradable COD, then the methane yield will be high because these microbes feed on the biodegradable COD. So here it is a figure for the anaerobic digester. So we can see that it has different parts. So the bottom part, it has space for ground sludge pipe and ground injection pipe. Next, it has mixing zone. At the bottom, the sludge after digestion, it settles at the bottom, and from here it is taken out from time to time. Above this, we have a mixing zone where mixing of the digester is done. Next is the sludge zone. In sludge zone, the active digestion takes place. Above that, it has fluid zone in which the supernatant accumulates, and this is taken out from the reactor. And there is also outlet for biogas, so that is the effluent gas. So there are two types of digester. It can be a low rate, the conventional digester, or it can be high rate digester. So the low rate conventional digester, in this intermittent mixing is done, and intermittent sludge feeding and withdrawal is done. And it has a detention time of generally 30 to 60 days, that is 1 to 2 months, and the feeding rate is also less as compared to the high rate digesters. And in the high rate digesters, continuous mixing is done so that the whole sludge and the feed inside the reactor is homogenized. It can be a continuous or intermittent sludge feeding and sludge withdrawal. And here the detention time is very less. We can say it is, we can see here it is generally less than or equal to 15 days, which is much lower than the low rate digesters. Also, the most digesters are heated and operated in mesophilic range, and are made of concrete or steel. So we have seen previously that with increase in temperature, the yield of the biogas can be increased, so that these reactors are generally heated to maintain the optimum temperature. So here some of the digestion or digester operation is given, like most digesters are operated in the temperature range of 30 to 38 degrees centigrades. So the gas which is produced, which it can be further used to for heating the digester, the optimum pH range is between 7 to 7.2, and it is maintained by properly seeding fresh added sludge and not excessively withdrawing sludge. Generally 3 to 5% of the dry solid waste weight in digester is decanted every time or on daily basis or whatever the cycle in which the sludge is taken out from the digester. Next is to compact acidification. Lime can be added in the reactor to increase the pH. And heavy metals may inhibit the digestion process. It can retard the growth of the microbes, so they are eliminated before feeding. Next is the supernatant liquor in the water, which is released after digestion. It can have high BOD and solids contained, so they are generally sent at the start of the treatment plant if sludge is being digested or some other feed is digested. So what happens during anaerobic digestion, complex organic compounds are converted to simpler ones. This is the hydrolysis step, and after hydrolysis, acidogenesis takes place, in which the simpler organic compounds are converted into acid. So the acidogenic reactions, for this the favorable pH is less than 6. It is around 5.5 to 5.9. The acidogenic bacteria thrives at the slightly acidic pH, but the methanogenic bacteria, these thrive at neutral pH or slightly acidic pH. So if and we have seen that the methanogens they are very sensitive to, and they have very slow growth rate. So to increase the efficiency of the reactors, two reactors can be used. In first reactor, hydrolysis and acidogenic reactions takes place, where the pH can be lower than 6, and in the second reactor, methanogenesis takes place, in which the pH is around neutral or slightly basic, so that proper acidogenesis takes place, and also proper methanogenesis takes place, and we get optimum yield of the biogas. So here is the schematic of two-phase digester model. Here we can see that acid reactor is different and methane reactor is different. So in two-step anaerobic digestion, main biochemical reaction in liquefaction is the main biochemical reaction that takes place in the first reactor is the liquefaction of organic solids, digestion of the soluble organic materials, and it has usually high rate digester with fixed cover and continuous mixing. And in the second stage, it is generally a conventional low rate digester with a floating cover and intermittent mixing. So the main process which takes place in the second stage is the supernatant separation, that we have to separate the water from the feed. Next is the gas storage. Whatever gas is produced in the reactor, it needs to be stored, and digested sludge storage, which is stored at the bottom of the reactor. So also the biogas which is produced, we have seen in the, I think, first slide only, that it has some other impurities like water, hydrogen sulfide, CO2 and dust, which need to be removed. Otherwise it can cause some operational problems during energy generation. So these are some of the methods for removal of moisture from biogas. Like we can follow condensation method or demister, moisture trap, or we can use silica and all. So next is the sludge volume. So the volume of digesting sludge in a digester, it will be function of volume of fresh sludge which is added daily, volume of digested sludge produced daily, and the required digestion time. So the volume of digesting sludge will be function of all these three. And from some empirical evidence and some experiments, it has been seen that if supernatant is removed from a batch of digesting sludge as it is produced, then volume of remaining digested sludge versus digestion time is a parabolic function, and we get some that the average volume will be V1 minus 2 by 3 V1 minus V2, where V1 is the volume of fresh sludge added and V2 is the volume of digested sludge produced, and V average is the average volume of the digesting sludge. So the total volume of the sludge in the reactor, it will be average volume and the V2, that is the volume of digested sludge, into their respective retention times. And in the anaerobic digester, the bottom half of the digester is normally sludge, and the top half is used for water, that is supernatant, and biogas. So the total volume of the digester is 2 times the sludge volume. So let's go to the next one. So this is the last lecture for anaerobic digester. Here the design of anaerobic digester is discussed. So here we can see that if it is a two-step digester, then it will have a primary first digester where acetogenesis and hydrolysis takes place, and there will be secondary reactor where methanogenesis takes place. And the factors which influence the plant performance are solid content, carbon to nitrogen ratio, temperature, pH, and retention time. We have seen this in the previous lecture. So again, here is I think we have seen this in the previous lecture itself. So here it is a graph which shows the effect of temperature on the biogas produced with temperature. So we can see here that the green one is 8 degrees centigrade. So we can see that with increase in the temperature, the biogas production has increased in all the types of uh, feed which is being used. So around 35 degrees centigrade is generally maintained in the anaerobic digesters. So here it is the carbon to nitrogen ratio. So generally carbon to nitrogen ratio of 20 to 30 is to 1 is maintained in the reactor. Temperature is the room temperature, around 20, room temperature, and it is generally in the mesophilic range. Next is pH is neutral, retention time is 60 to 90 days, or it can depend upon the type of the reactor. And for mesophilic digestion, it is greater than 20 days. So here it is a cross section of the digester. So the bottom layer will be the sludge layer volume. Above that it is the active digestion volume. Above that it is the gas storage volume, and top above that it is the gas collection volume. So the overall total volume of the digester will be sum of all these four. So here it is the dimension of the reactor. So it consists of three parts. Top uh, it is it consists of three volume actually, V1, V3 and V2. V3 is the cylindrical part, V1 and V2 are the parabolic part, and these are the nomenclature which are being used. So here there is difference between cross section and dimension. In dimension we find out the dimension of the each of we find out dimension of these three volumes, that is V1, V2 and V3, and if you see the cross section, it consists of five parts because it is used for five different purposes. So here is a formula which are used to find out the different dimensions of the reactor, and there was a numerical based on that we will see in the assignment questions. So I think that's it. So next is the third lecture. The third lecture is, sorry, this one is the fourth one. It is on introduction to microbial fuel cells. So the different types of cells related to energy. First is the galvanic cell which converts the chemical energy to electrical energy. Next is the electrolytic cell. It converts electrical energy into chemical free energy. Next is the batteries. Batteries are the devices which carry out these conversions. Generally, in batteries, there are some sort of chemicals, and the chemical reactions from these convert the chemical energy into electrical energy. Next is the fuel cell. So, fuel cell is a electrochemical device in which the free energy of a chemical reaction is converted to electrical energy. The reactants are supplied from a external source as they are consumed, and no combustion and no NOx emission, because generally during combustion air is used and air has nitrogen. Also the feed which is being used, it has nitrogen, and NOx emissions are there in the product side. Next is the microbial fuel cell. So microbial fuel cells are type of fuel cell in which the chemical reaction which takes place in the fuel cell a cell are replaced by the microbial reaction. Here microbes are used to do the to perform the reactions, and the organic load of the wastewater is used as fuel. So here we can see a fuel cell. So this is the fuel cell. We can see it has an anode electrode and cathode electrode. So generally hydrogen are fed towards the anode where oxidation takes place, that is H plus and electrons is produced, and reduction takes place on the cathode side where oxygen is fed. So there is a membrane which only allows the flow of hydrogen ions. So the from oxidation of hydrogen H plus is produced. So this H plus flows from the anode side to the cathode side through by this membrane, and the electrons these are transferred from the anode side to cathode side by external circuit, and we know that flow of electrons gives us current. So through this electricity is produced. So here it is a microbial fuel cell. We can see that this is some sort general equation used for organic matter. So it will be oxidized to CO2 and it will give H plus and electrons. And on the cathodic chamber, oxygen will be reduced and it will form H2O and it will consume H plus. So there will be some sort of semi-permeable membrane that will allows the flow of hydrogen, and electrons will be transferred through external circuit and through the organic matter present in the wastewater, and through microbes this circuit will be completed and it will give us current or energy. So here mechanism of the electron transfer is discussed. So please go through these. So here there are some of the design of microbial fuel cell, that what are the materials used for different parts, like what material is used for anode, cathode and proton exchange system, and electrolyte, electrode catalyst. So here different types of microorganisms are given with their different pathways. So please go through all these. So So there can be two types of the culture which are used in microbial fuel cell. It can be a pure culture or it can be a mixed culture. Generally, mixed bacterial culture gives are more resistant to disturbance, and their substrate consumption rate is also high. And also, it in the pure culture, bacterial culture, there will be very high risk of contamination due to very high risk of contamination from the undesirable bacteria. So, as compared to pure bacterial culture, mixed bacterial culture have higher output. So these are the some of the parameters which affect the MFC operation, and please go through all these. So there will be obviously some losses. Please go through these slides. So let's go to the last lecture of week 6. So this is on fermentation. So the first part of fermentation is in week 6 and the next part will be on week 7. So what is fermentation? It is a metabolic process which converts sugar or acid to alcohol or gases. So the organic waste or biomass, it can either be digested anaerobically. We have seen the anaerobic digestion. It can be fermented to ethanol or like alcohol, or also the organic waste can be gasified to produce syngas, and that syngas can be further fermented to produce alcohol. And ethanol, it can be used as a blend with gasoline since it has high calorific value. And butanol, it can be used in pure form or it can be blended with gasoline at higher concentration because it has higher heating value. Next is fermentation is a series of chemical reactions which convert sugar to ethanol in presence of suitable microbial strain which feed on the sugar. And the end product is alcohol, that here it is ethanol. And here reaction is given for compound containing 6 carbon, and it is converted to ethanol. And generally is yeast is used for hexose, like 6 carbon containing compounds. So we can say depending upon the number of carbon atoms, it can be hexose because the compound has 6 carbon, and if it is 5 carbon, then it will be pentose and so on. So here we can see a flow chart for production of ethanol from a feedstock. So the sugary crops can be processed and fermented to produce the ethanol, but after processing, the fermented mash has very less ethanol. It has around 4 to 10% ethanol. So it is distillated and we get 95 to 96% ethanol. But for mixing the ethanol with gasoline, we need to remove the water content, and so dehydration is done where water is removed by following different methods, and we get around 99.5% of the ethanol. So here it is that the concentration of ethanol in the fermented mash can attain a value of around 4 to 10%. Therefore, distillation is done to get around 95 to 96%, and after that, it is further dehydrated to get around 99.5% of the ethanol. So, sugar can be fermented directly, and if it has some starch, then first starch is hydrolyzed to sugar, and if the feed is cellulose, then it is also hydrolyzed to sugar and then fermented. And as we can see that the pre-processing steps increase as we go to complex biomasses. So sugar will have no pre-treatment, then starch will have more pre-treatment than sugar, and then lignocellulosic biomass will have more intense pre-treatment. So here production of ethanol from starchy crop is given. So first step is cleaning and steeping of the grains. Next is grinding. After grinding, germ at the center of the corn is separated, and then it is washed and filtered. Then gluten is separated, and then it is cooked. And then the step 5 is cooking, where starch is converted to sugar, and finally fermentation takes place, and distillation and dehydration is done to get the alcohol. So this is the wet process, and next is the dry process. So in dry process, first cleaning is done, then milling, then liquefaction, saccharification, where starch is converted to sugar, then fermentation, followed by distillation and dehydration. So please go through these slides, where ethanol is produced from lignocellulosic biomass. So it will also go through some pre-treatment processes, and then it can be used for alcohol production. So here some of the common lignocellulosic biomasses are given, like MSW, that is the municipal solid waste. So we can use the food waste, paper or sorted refuse, and this can be used for fermentation after proper pre-treatment. So here the pre-treatments are given, so please go through these. So here different techniques for pre-treatment are given, like we can use physical treatment like cutting and grinding, which we can use chemical treatment, we can use acids and alcohol, we can use a combination of physical and chemical treatments, that is physicochemical treatments, and also we can use biological treatments by using some microbes. Next is detoxification. So during fermentation, along with ethanol and butanol, lot of other unwanted products are formed, and if their concentration are not controlled, then they can be harmful for the microbes which are performing fermentation. So their concentration need to be controlled, and some sort of inhibitors or and some controlling operating conditions are controlled to control the concentration of these byproducts. So these were the parts in the fermentation. So these were the 5 lectures in week 5. So now we will move to the questions. So the first question is, which of the following is not a primary product of anaerobic digestion? First is hydrogen, next is biofertilizer, C is reusable water, or D bio-oil. So I'll wait for 2 minutes and then we will discuss the answer. So we have seen in the first lecture itself of the anaerobic digestion that we get hydrogen, we get biofertilizer, we get reusable water, we get biogas, hydrogen, biofertilizer, reusable water, but we do not get any bio oil from anaerobic digestion. So we get bio oil by gasification and pyrolysis of the solid waste, but we do not get bio oil in anaerobic digestion. So D is the correct answer. So let's go to the next question. So the question is, match the following major component of biogas with their appropriate percentage. So biogas consists of methane, carbon dioxide, oxygen, water and others. So we have been given carbon dioxide, methane, nitrogen, oxygen. So we have to find out what is the percentage of these gases compounds in the biogas. So again, we will discuss the answer after 2 minutes. So we can see from this slide the major components of biogas. It has 50 to 70% methane, 25 to 50% CO2, 0 to 10% nitrogen, and hydrogen is around 0 to 1%. Hydrogen sulfide is 0 to 3%, and oxygen is 0 to 1%. So carbon dioxide is 25 to 50%, so it will be S. For methane, it is 50 to 70%. So after S, it will be T. Nitrogen is 0 to 10%. So it will be R. And last oxygen is 0 to 1%. So A is the correct answer. So let's move to the next part. Generally, dash is the rate limiting step involved in methane formation. First is acetogenesis, next is acetogenesis, C is methanogenesis, or D hydrolysis. So again we will wait for 2 minutes and then we will see which is the correct answer. So we have seen in the slides that growth yield of methanogenic bacteria is very slow. So generally methanogenesis is the rate limiting step. So but if lignocellulosic biomass are used as feed, then hydrolysis can also be the rate limiting step. But here no condition is given. So methanogenesis will be the rate limiting step. So C is the correct answer. So let's move to the next question. So question is, in anaerobic digestion, the presence of lignin decreases the rate of A. Acidogenesis, B. Acetogenesis, C. Methanogenesis, or D. Hydrolysis. So again we'll see the answer after 2 minutes. So we have seen So we have seen that if the feedstock has lignin, the hydrolysis will be the rate limiting step because lignin degrade very slowly. So D is the correct answer. So let's move to the next question. So the question is theoretical substrate conversion rate per unit reactor volume can be estimated as. So it was there in the slide. So we can see that the theoretical substrate conversion rate per unit reactant volume can be estimated using this formula, where R is the substrate converted per liquid volume at hydraulic retention time of theta, S naught is the substrate concentration in the feed, mu max is the maximum specific growth rate, Ks is saturation constant or substrate concentration at which the specific growth rate is half of the mu max, and this is the reaction which is used. So this is the formula which is used to calculate substrate conversion rate per unit reactor volume. So D is the correct answer. Sorry, not D. C is the correct answer. So the photo is really blurred. So A is the correct answer. So let's move to the next question. So the question is match the following waste with their typical methane content. So three types of waste are given and we have to match with the methane content which they produce during anaerobic digestion. So again we will see the solution after 2 minutes. So let's see how to solve this question. So basically we have to refer the table which was given in the slides that grass can produce around 84% of the methane in the biogas. So for grass S and from corn silage around 52% of the biogas is methane, and for poultry litter we can see 68% is the methane gas. So S, Q and R. So B is the correct answer. Don't know what happened. So the next question is, galvanic cell is used to convert A, electrical energy into chemical free energy, B, chemical energy to electrical energy, C, mechanical energy to electrical energy, or D, none of the above. So we have seen the definition of a galvanic cell in the second last lecture, which was about microbial fuel cell. So we will see the answer after 2 minutes. So galvanic cell is used to convert. So we can see that galvanic cell, it converts chemical energy to electrical energy. So B is the correct answer. So let's move to the next question. So the question is, dash is a metabolic process that converts sugar to acid, gas or alcohol. So the options are fermentation, anaerobic digestion, pyrolysis or gasification. So we'll see the answer in 2 minutes. So fermentation is the metabolic process that converts sugar to acid, gases or alcohol. So A is the correct answer. So let's go to the next question. So depending on the operating condition and strain of yeast used, fermented mash can attain a level of around dash percentage of ethanol concentration. So it is 4 to 10%, 10 to 20%, 20 to 25% or D 20 to 30%. So again we'll see the answer for this question in next 2 minutes. So ethanol concentration in the fermented mash is around 4 to 10%, which is further after distillation it increases to 95 to 96%, and after dehydration it increases to 99.5%. So A is the correct answer. So let's go to the next question. So the question is, the type of losses in microwave fuel cell caused by uneven depletion of reagents in electrolyte is called as A. Ohmic losses, B. Activation losses, C. Bacterial metabolic losses, or D. Concentration losses. So we have seen that we have seen different types of losses in an MFC, and we have to find out what is the loss called caused by uneven depletion of reagents in electrolytes. So again we will see the answer in 2 minutes. So we can see that concentration loss is because of uneven depletion of reagents in the electrolyte, which causes concentration boundary gradients in the boundary layers, and this reduces the efficiency. So D is the correct answer. Let's go to the next question. So the question is identify the different zones in anaerobic digester. So we have to identify what is A, B and C in this figure. So here we can see a picture of an anaerobic digester. We can see that C is the mixing zone. Let's start with A. A is the fluid zone where the supernatant is in the digester. B is the sludge zone where the digestion of the this is the active zone where digestion of the feed take place, that is the sludge zone. And C is the mixing zone. So A is the fluid zone, B is the sludge zone, and C is the mixing zone. So C is the correct answer. So let's move to the next question. So we have to identify A, B, and C in the fuel cell. So here it is the picture. We have to identify what is A, what is B, and what is C. We will again see the answer in 2 minutes. So in the fuel cell, where hydrogen is fed, it is the anode where oxidation takes place, and where or air is fed, it is the cathode where reduction takes place. So, and also there is a permeable membrane through which only H plus moves from the anode side to the cathode electrode. So C is the proton exchange membrane, A is the anode, and B is the cathode. So A is the correct answer. So let's move to the next question. So this question is based on determination of volume of the digester chamber. So the question is, determine the volume of the digester chamber for the biogas production from the cow dung of 20 cows having body weight of 200 kg each. Assume the temperature is 30 degrees centigrade, the solid content of cow dung is 20%, and HRT is 40 days. One cow produces 15 kg cow dung daily. Consider favorable condition is 8% concentration of total solid. So we have to find out what is the volume of the digester chamber. So we will wait for 2 minutes and then we'll see how to solve this question. So let's see first basics about this question. So the total volume of a digester consists of VCO, that is the collected volume, gas collection volume, VGST, which is the gas storage volume, VD is the active digestion volume, and VSL is the sludge layer volume. And here we can see that VGST plus VD is 0.8 times of V, that is the total volume. So let's start here. And we have to find out the digester chamber, volume of the digester chamber. So here in the question, we will first find out how much discharge is from the cows per day. So total discharge it will be number of cows which is 20 into how much cow produces cow dung in one day. So one cow produces around 15 kg of cow dung. So 20 into 15. So 300 kg per day is the discharge of the cow dung. Next, we will find out the solid content. So we can see here that the solid content of cow dung is 20%. So total solid content will be 20% of the discharge. So it will be 0.2 into 300. So it will be 60 kg. Next, it is given in the question itself in that considerable favorable condition is that we need to have 8% concentration of the total solid. And we can see now, so we have 60 kg of the total solid content. So we have to see how much we have to dilute this. So 8% solid content means that 8% TS means we have 8 kg of solid in 100 kg of total influent. So for 60 kg of solid, we will need 100 by 8 into 60 kg of the influent, which is coming out to be 750 kg. So total influent required is 750 kg. And working volume of digester is given as Q into HRT, where Q is the rate, discharge rate, or we can see how much influent we have to feed per day, and HRT is the retention time. So we have 750 kg influent per day, and HRT is given in the question as 40 days into 40. So the working volume is coming out to be 30,000, it will be 30,000 kg. So, and we know that 1000 kg is equals to 1 meter cube. So 30,000 kg will be 30 meter cube. But now we have calculated the working volume of the digester, and we have to find out the volume of the digester chamber, and it is generally taken as 0.8 times of the working volume. So working volume is taken as 0.8 times of the digester volume, where V is the digester volume. So digester volume will be working volume divided by 0.8. So let's see what we are getting. So we are getting 37.5 meter cubes as the digester volume. So this question was very straightforward. Similar question was also there in the a similar question was also there in the design third lecture of the anaerobic digester. So what we have to do, we have to find out the total discharge of how much cow dung we are getting per day, and then we have calculated the total solid content, and based on the favorable condition for the reactor, we have converted that into kg in total concentration. We have find out the total solid contents in the cow dung, and based on the favorable condition, we have find out how much influent we will be feeding in the reactor. So it's coming out to be 75 kg influent per day, and the working volume of digester is given by uh, feeding rate into HRT. So it is coming out to be 30,000 kg, and since 1000 kg is equals to 1 meter cube, we will convert this into meter cube, and we are getting the working volume of 30 meter cube. And we know that, and also we know that working volume is 0.8 times of the digester volume. So we will get the digester volume by dividing the working volume by 0.8. So which is coming out to be 37.5, which is nearer to 40. So B will be the correct answer. So these were the questions for week 6. Next, we have two more problem solving session based on week 7 and 8. And then those who have registered for the exam, they will be giving the final exam. So thank you so much. And we will again meet on Saturday from 3 to 5 p.m. So, and we will discuss the materials, lecture materials and questions based on week 7. So thank you so much for joining. Have a nice day.